Hello, everyone, and welcome to our Q&A session with Dr. Deborah and Dr. Andrea. First of all, I would like to thank everyone for being here with us today. And on the behalf of our committee, I would like to um, say that we hope you guys are enjoying our Congress so far. Dr. Andrea and Dr. Deborah, would you like to join me for this flash talk?
Hi.
Hello, Dr. Deborah. It's so nice to meet you. I would like to say what a pleasure it is to have you here with us today. Thank you so much for accepting our invitation to the Congress. I'm very excited to read all the questions and hopefully you can answer them for us. Welcome, Dr. Andrea. Same as well. Nice to meet you. It's very nice to have you here with us today, answering all the questions that we have from the audience. Welcome.
Thank you so much for the invitation, and I'm happy to be here with you.
Oh, it's our pleasure. We're very excited to have you guys with us today. Unfortunately, Dr. Amir and Shivesh couldn't be here with us today, but we will be sending out all of the questions personally to them so they can hopefully answer them by email, and we'll be in contact with you as soon as we can. But okay, let's get started with many questions that we have from the audience. The first question is from Andre. This question is directed to Dr. Deborah, but this is a flash talk, so feel free to join the conversation, Dr. Deborah. Andre says, Dr. Deborah, given the positive outcomes observed in facilitating cell growth and attachment with the addition of CNC or CNF to the hydrogel, what considerations arise in the optimizing and concentration and distribution of these cellulose nanomaterials for maximal biocompatibility while maintaining the material's structure integrity? Very long question. I hope you understood that, Dr. Deborah.
First of all, thank you for the invitation to be here. Actually, it was a really long question, but regarding the first part of the question, I acknowledge indeed it was. I have a really good results. We are actually now trying to put some efforts to validate this strategy in the lab in mice models to see if we can have the same results that we have in vitro and also in a model of CAM in vivo. In the first preliminary results, we have outstanding results. We also observed the reduction of the tumor, but we are currently analyzing all the samples and all the results to be sure that we have really good results.
Okay, thank you, Dr. Deborah. We have another question for you as well, following the respective of the presentation. This one is from Anna. Thank you, Anna, for your question. Dr. Deborah, what are the main disadvantages of exosomes and how can we circumvent them?
So actually, we are also trying. We are also working with some. We are also doing some experiments to circumvent some of the problems that the exosomes face. For instance, we are now working on trying to modify at the surface these exosomes with some sort of ligands, for instance, with peptides and aptamers, to confer more specificity and more selectivity, in this case for the triple negative breast cancer cells, to try to circumvent, as I said, some of the difficulties of the targeting of these exosomes. One of the another main problem that exosomes face is it is a field that is starring, so there is some there is a lack of sometimes of characterization methods but they are now currently being developed. So I think that in a couple of years we will have really good methods and novel equipments that can be used and be useful for this characterization of exosomes.
Perfect. Thank you, Dr. Deborah. Very promising work. Very, very great. Okay, next question is for Dr. Andrea. It's also from Andre. He says, how do you determine the optimal proportion of structural material, such as gelma, alterate, within the hydrogel fiber to achieve a balance between mechanical stability and the incorporation of microchannels for nutrient transfer?
Okay, so thank you so much for the question. Basically, I characterize the mechanical properties through tensile assays, and I change the proportion of the structural material, in this case, high viscosity alginate. And I design different variations of this proportion inside the fiber, and I printed different types of fiber through the chaotic printing strategy that was shown in the presentation. And after that, I did a tensile essays, 5 repetitions for each type of variation. And after that, in base of the stress strain curve that which I obtained, I extracted from the data the 3 parameters: the elastic modulus in kilopascals, strain at break, and stress at break. And I did a ANOVA analysis to determine if there were relevant differences between the values of these 3 variables, right? And so after all this analysis, I found out that the optimal proportion was to have at least 3 out of 8 inlets with a high viscosity material. And even if we have higher elastic modulus values with more amount of high viscosity alginate, you should also remember that the final objective of the fibers is to do grows myoblasts on them, and myoblasts don't like a very stiff material like high viscosity alginate. So it was basically all this mechanical assessment, but also the criteria in which I base which type of fiber variation to choose was also considering that the fiber cannot be 100% high viscosity alginate. So basically, that's how I optimize the proportion of structural material.
Okay, very good. Thank you so much, Dr. Andrea, for answering. Our last question for the 2 of you. We have a few more questions, but they will be sent out. Unfortunately, the other candidates couldn't be here with us, so we will end the session a little early. Before we say our goodbyes, is there anything that you would like to say, both of you? Okay, great. So thank you so much for being here with us today. It was an honor to actually present these questions to you guys and say what a pleasure it is. And I hope that you guys keep up the great work, and I hope to see you guys soon with new research in the next congresses to come. Okay, see you guys soon. Have a good night.
Thank you. Have a good night.
Thank you so much. Have a good night.
Thanks.